This is a production of Cornell University.
I've been in CEE for 7 years now, coming from a postdoc in environmental biogeochemistry at EPFL in Switzerland, which I'll talk a little bit about. And so, the topic is on biogeochemistry of arsenic in rice paddy soils. I was telling Lou, I sent that title in the summer. I think I was still in denial about the semester starting, so I just sent a very generic title because I didn't want to decide what I wanted to talk about. So we have a little bit of a focusing sort of subtitle here on the role of dissolved organic matter in microbial arsenic transformations. So my group over in civil and environmental engineering is on biogeochemistry and ecosystem engineering, and we have kind of like two broad research themes that we pursue in my lab. One is on nature-based systems for controlling non-point source nitrogen from agriculture, also some urban stormwater. So this is things like constructed wetlands, biofiltration. This is a wood chip bioreactor at a farm near Cornell. And the other research area that my group pursues is in the microbial biogeochemistry of arsenic. And a lot of that work is motivated by the problem of arsenic uptake into rice and trying to understand those processes better, especially from a below ground perspective. Like, what are the soil biogeochemical processes that are controlling the speciation, toxicity, and fate of arsenic in rice paddy soil environments? And so sometimes people, and I'll be talking about this today. So sometimes people ask you how, especially from environmental engineering, that I get involved working with rice. So as a graduate student here, I was working in wetlands in New Jersey. So New Jersey wetlands are a great place to study pollutant dynamics in wetland environments. And so here's a picture. We were doing some work in the wintertime in the New Jersey meadowlands, which you see if you sort of drive on any of the highways around sort of northern New Jersey. And a lot of what we were doing was, thinking about actually focusing in on the wetland rhizosphere, the release of oxygen from parenchymal wetland roots into anaerobic wetland soils, the formation of these oxic-anoxic interfaces in the subsurface, and how that affects cycling of redox-sensitive metals like iron and the coupling of that to other biogeochemical processes. And we were also interested in how this sort of gas transfer conduit can also facilitate the release of methane and nitrous oxide. So then after that, I did what one does when they're interested in aquatic chemistry. I moved to Switzerland for a postdoc at EPFL. And as I was discussing projects with my postdoctoral advisor, Rizlan Bernier-Letmani, who's actually a Cornell undergraduate alum, she mentioned that they were just beginning a new research collaboration and partnership with some groups in Vietnam. And this was part of a like Francophone research initiative called RESCIF. Here, why was Vietnam involved in this Francophone research network? Because of the sort of older colonial history in Vietnam. And part of this sort of this Asian Center of Water Research. So this is a sort of a map that shows sort of this like research network connecting different Francophone research institutions, including ones in Vietnam. And that's how sort of we go from kind of like cold New Jersey, snowy wetlands to sort of tropical Mekong Delta rice paddy environment, sort of like connected by the theme of sort of biogeochemical processes and flooded soil environments. Okay. And of course, so talking about rice and arsenic, a lot of that work that I was interested in starting in the mid 2010s, a lot of that came out of important work that happened in this department, okay, in the group of John Duxbury, which was some of the first work to identify the problem of high arsenic concentrations in rice, but then also importantly, identified that we have to look beyond total arsenic and we have to think about arsenic speciation, okay, to think about what are the concentrations of inorganic arsenic species and the concentrations of methylated arsenic species. So this is monomethyl arsenic, where one of the hydroxyls is substituted by one methyl group and then a di-DMA. I'll be talking about DMA a lot, a dimethyl arsenic with two methyl groups, okay? And what was found here was that rice a larger fraction of methylated arsenic species compared to inorganic arsenic species. And that using market basket surveys, there could be a large variability in the methylated arsenic concentration, but the inorganic arsenic concentration seems to not change very much. Okay, so this is really important early work motivating why we should try to understand the dynamics of these methylated species better in the soil, because the rice plants themselves are not capable of methylating arsenic. The methylated arsenic is formed primarily from microbial transformations. Okay? There are some organoarsenic pesticides which are used, which can produce some of these methylated species, but for the most part, we think these are produced by microbial biomethylation reactions that happen in soil. Okay, so there are three parts in the talk today. I'll start in the first part with a kind of bigger overview of the problem of arsenic in rice paddy soils and what some practical efforts are to deal with it. And this is meant partially to provide just a general overview for anyone who's not familiar with the topic. And then we'll talk about some.
I think the mic may have died.
Battery. Oh yeah, this has a red instead of green. Okay.
Will it become green or it stays red?
It's red.
We always have a lot of fun with the type of problems. This one is a range as well.
Okay. Maybe some. Can you hear me now? Anyone? I guess here. Okay, great. Thank you. Thanks for the Zoom comment there. So we'll then dive into some more focused research here on the effects of dissolved organic matter on arsenic methylation, and then we'll wrap up with a brief discussion of arsenic demethylation. Okay. So this is a X-ray fluorescence map of some rice paddy soil from Arkansas, and it shows the sort of the co-locations of arsenic, iron, and manganese. Okay, so co-locations of arsenic and iron have this yellow color, and so what you'll see is that there's quite a bit of yellow, especially around the edges of some of these mineral grains. Okay, and what that's telling us, and this is something that's been known for some time, is that arsenic strongly absorbs onto the surfaces of iron oxide. Okay, so we want to understand sort of processes regulating the solubility of arsenic and mobility and bioavailability of arsenic. We have to think about iron. Okay, and so we also went in and did some Zane's spectra on some hotspots here. So you can see some of these POIs, like points of interest, like 2, 3, 6, around these yellow colors. You can see that so this is some arsenic Zanes, that the sort of the Zane spectrum is very similar to a ferrihydrite bound arsenic 5. Okay, so arsenic 5 is a really important host for arsenic in soil. You'll also see in some of these cases here, where there is these, you can kind of see them, maybe these like really small red dots, right? Like super hot for arsenic, but not associated with iron or manganese. A lot of these have a spectrum similar to arsenopyrite, okay, or like a iron arsenic sulfide mineral, and that's because arsenic also has a high affinity for sulfur. Okay, so what can often happen is there'll be this two-phase process if the soil goes from oxic conditions. If you think about a well-drained oxic soil, arsenic tends not to be mobile, it's bound onto iron oxide surfaces. If the soil is flooded and becomes anaerobic, we have reductive dissolution of iron oxide, release of iron too, and also mobilization. So it's very common that you see this correlation between dissolved iron and dissolved arsenic in
wetland or rice paddy soil pore water because the reductive dissolution of iron controls the release of that arsenic. Okay. At the same time, if you have systems high in sulfate that become sulfidic, you get sulfide, then the reaction of sulfide, arsenite, and iron 2 to produce arsenopyrite, or also to produce like mechanoite iron sulfide minerals, and arsenic also stores into the surface. So, sort of like big picture, there's a lot of recognition that iron is important for arsenic dynamics, sulfur is important for arsenic mobility, and so if you have a rice paddy soil that's flooded, this happens, and we get mobile and bioavailable arsenic, which is one reason why, the principal reason why rice is so good at taking up arsenic, because it's grown in flooded soil conditions. Okay. So it's known that this is a problem. So when I first started at Cornell, I had a collaborative project with the Dale Bunkers National Rice Research Center in eastern Arkansas, which is where most of the rice in the United States is grown, on some of their efforts to basically try to control the bioavailability of arsenic through basically controlling irrigation. Okay, so rather than grow rice continuously in flooded conditions, do something called alternate wetting and drying, which is to periodically drain the soils, oxidize the soil, sort of like reverse this process. Okay. And then what we did is we collected pore water samples to monitor the pore water geochemistry during these alternate wetting and drying experiments. We also collected soil samples to take to the synchrotron, which is where we got the first dimension. So here's just an example of what we see. We have 3 fields. This shows the volumetric water content. This field was flooded for the entire season. This field experienced 1 dry down before being reflooded. This field experienced 2 dry downs. And so what you can see is if we look at the iron concentrations, we see this increase in the iron concentration, continuously flooded field because there are these anaerobic throughout the entire growing season. Okay. Error bars are big, all right, because we're looking at sort of like in situ pore water samples, a lot of spatial variability. The trends are pretty clear. If we look at this 1 drain field, you can see the iron is increasing, and then this dry down is pretty effectively at knocking those dissolved iron concentrations down. And then they do start to accumulate a little afterwards. And then you see the same pattern here where our iron increases, the dry down decreases it, increases and increases. So you can effectively control basically iron redox reactions through these very sort of practical water level treatments of the paddy soils. Is it effective for arsenic? Yeah, so these show pore water arsenic concentrations, total arsenic concentrations. So we can see here in the permanently flooded field, arsenic concentrations are relatively high. There's a lot of variability, but we do see a high mean. In the soil that's drained once, we see arsenic is increasing, and then it's very effectively controlled through even just this 1 mid-season drain event. It does start to come up here a little bit at the end. Okay, so from a sort of below ground perspective, we see that these water level treatments are effective at controlling arsenic concentrations. But ultimately, we care about like how much is getting into the rice, right? That's sort of like a practical endpoint that we are concerned with. So we also sampled the rice grains and performed arsenic speciation analysis of the rice grains. And so here are 5 different rice cultivars, okay, that were selected by our USDA collaborators in some of their like breeding efforts to also try to use breeding to select for strains that are excluders of arsenic. And so the red here shows the DMA, that methylated arsenic concentration, and the sort of teal color here shows the inorganic arsenic concentration. And so what we see is that, and this is sort of the 0 drains, continuously saturated, 1 drain, 2 drains. So the drains are effective at decreasing the total arsenic concentrations in some cases. Okay, so that's good news. But the less good news is that they're only effective at really changing the methylated arsenic concentrations, while the inorganic arsenic concentrations are fairly stable, okay, across all of these different drainage treatments and even across the different cultivars, which is interesting. And this is actually sort of consistent with what we saw in that paper from John Duxbury's group in 2008, using a market basket survey, that we see variability in DMA, while inorganic arsenic is relatively constant. Here we see something similar when we're looking at different cultivars, different irrigation treatments, but from the same sort of set of experiments. Okay. So we see that AWD did not have a significant impact on arsenic levels. Inorganic arsenic is actually the more, so this is a little bit controversial, but inorganic arsenic is thought to be the more toxic form of arsenic. So what we have in terms of like sort of nascent regulations about arsenic in rice, especially which is primarily focused on arsenic concentrations in infant foods, rice crackers, things like that, have focused on only inorganic arsenic. Okay, and this is similar to the regulatory structures in China or also in Europe, where the regulations focus only on inorganic arsenic because that's not to be used. Okay. So basically, if there's a concern or the goal is to control inorganic arsenic concentrations, for the most part, alternate wetting and drying is not very effective. But it does raise questions about like what's happening during these dry down events, so that inorganic arsenic is stable, while
methylated arsenic concentrations
are highly variable. I also want to mention briefly that there are a variety of objectives with alternate wetting and drying. One is control of arsenic. Another is control of methane. Okay, rice paddies are really significant global sources of methane emissions, and these wetting and drying cycles are also effective at significant emissions. Rice paddies, they also save water. Okay, so there's a variety of reasons why we might want to introduce this type of alternate
wetting and drying irrigation practice beyond the motivations for arsenic. Okay.
So there are also large-scale variations in sort of methylated arsenic fractions regionally. So if you look at here in the US grown rice tends to have a higher methylated arsenic fraction. Rice grown in Asia tends to have a lower methylated arsenic fraction. These patterns are not well understood at all. Okay, and this is from a paper that was 10 years old, but in the past decade there hasn't been much actually advancement in understanding.
Why?
So for the rest of the talk, we'll sort of do like kind of a deep focus dive on this process here. How is inorganic arsenic converted into methylated arsenic species in the rice paddy rhizosphere? What are the factors that control this microbial biomethylation process? That is basically controlling how much DMA and how much arsenic are available for uptake into the. And so we'll take a solidly sort of
Low ground.
Look at these methylation reactions. Okay.
So a lot of the work
I'll talk about now is work that my PhD student John has done. And so, as I mentioned, there's been a lot of research over the past 10 years, research using metabolic inhibition of different microbial metabolisms to try to link methylation to a specific sort of functional group. Like, is it related to sulfate reducers? Is there some relation to antigens? Whatever. That has not been particularly conclusive. Also a lot of work looking at RSM, the functional gene for methylation, RSM diversity. Like, we can learn things from RSM diversity. It turns out that RSM is extremely diverse. Okay, so it's hard to link the sort of the genetic potential for arsenic methylation to any specific sort of
microbial functional group. So what we did is we sort of
thought, okay, let's like sort of step back and think back to basics. How does arsenic methylation happen? Okay, RSM, the RSM enzyme that methylates arsenic, is an intracellular enzyme. So in order for arsenic to get methylated, it first has to get into microbial cells. Okay. How does arsenic get into cells? Okay, depending on the oxidation state of arsenic, arsenate can enter cells through phosphate transporters because of chemical similarities with arsenate and phosphate. Okay. But in
reducing systems, the predominant
form of arsenic is actually trivalent arsenite. And arsenite is neutral, has a pKa of around 9. So in most environmental soils, it's uncharged, and it enters cells through GLPF transporters. And GLPF transporters typically take up uncharged small carbon molecules like glycerol or glycine or sugar. Okay. So we decided to sort of think about, what's happening here, thinking about how arsenite gets into cells so that it can be methylated into DNA. Okay, and one of the tools that we've used is an arsenic biosensor. Okay, so an arsenic biosensor is engineered E. coli to produce fluorescent proteins when arsenic is taken up into cells. Okay, so you can use this to sort of, so this is a fluorescence microscope image of these biosensor cells. So you can see here these cells like lighting up in red. That's because they're expressing these fluorescent proteins because they have arsenic taken up inside the cell. This work has been done in collaboration with Alex Lawn at University of Ottawa, who's a microbiologist who developed these biosensor cells. And so basically we can measure the intensity of the fluorescence as a quantitative measure of how much arsenic is taken up into cells or how quickly arsenic is being taken up into cells. And we first started using this technique to understand, what role does dissolved organic matter play in complexing arsenic, possibly through sulfur ligands, possibly through colloid formation. And is that something that is limiting the bioavailability of arsenic so it can't get inside cells? So one application of this was to think about oxic and anoxic interfaces in soil that are produced when we have these wetting drying cycles, also these oxic and anoxic interfaces that can occur in the rhizosphere where there's oxygen release from roots into anaerobic soil environments, and the production of these sort of like Fe3 NOM or Fe2 NOM colloids, okay, which Hyun produced in the lab, and then filtered through a 0.2 micron filter, okay? And this is important because often we think about the filtered concentration as sort of like the dissolved fraction. This is the bioavailable fraction that passes through this 0.2 micron filter. And so what Hyun showed really well is that that's not really true at all. So we looked at different arsenic to carbon ratios. So the arsenic concentration fixed, so increasing concentrations of dissolved organic matter. And then also looking at the iron 3 to carbon ratio as iron 3 concentrations increased. And you can basically look at the size of the bubble as a measure of arsenic uptake, okay, using the bias. Where 100 is sort of like, what we see in the absence of any DOM or iron. And as we go to sort of higher DOM concentrations and also higher iron concentrations, we see the arsenic uptake increase. We see the arsenic uptake decrease, the amount of inhibition increase, until here we actually get to these points where at high iron 3 and high DOM, there's actually no arsenic uptake at all. So arsenic uptake is like completely suppressed at these conditions, even though the arsenic is still like in the dissolved fraction. Okay, which is a quite interesting result. So one process which might be limiting arsenic uptake into cells and transformation is sort of complexation of DOM, or complexation or partitioning onto iron 3 colloids, which are stabilized by the presence of dissolved organic matter. Okay. Rice paddies are very rich in dissolved organic matter. That's the reason why they're such important sources of methane. Okay. And so we also then were interested in the role of sulfur-rich DOM due to the role of thiol ligands in complexing arsenic. I mentioned earlier that arsenic has a high affinity for sulfur. Okay, so thiol ligands in dissolved organic matter have a very high affinity for arsenite. We also studied the model compound glutathione here. And so what we saw is across a range of arsenic concentrations, when we increased the thiol to arsenite ratio, we saw decreasing uptake of arsenic. Okay, so basically more DOM, less uptake. Okay, it's sort of consistent with what we saw in that previous figure. But one interesting thing here we saw here was this upper Mississippi River natural organic matter, which is a reverse osmosis isolate of river water from the Mississippi River, has a relatively high organic sulfur content, but it was able to achieve significant decreases in arsenic uptake at much lower thiol to arsenic ratios than glutathione, right? The work with glutathione told us that you need a very large excess of thiols to actually see a meaningful inhibition of arsenic uptake. We didn't need such a large excess in the presence of the Mississippi natural organic. Okay. So this suggests an inhibition mechanism other than arsenic complexation with thiol. Okay. So complexation plays a role here, but it might not be the whole. And so what might also be happening is there some like biological response here, as well as just sort of like a chemical complexation. And what we saw was that when we look at OD600, so we look at cell growth, the cell growth is stimulated in the presence of the Mississippi NOM when it's not really stimulated in the presence of the okay. So this is telling us that there's biolabile carbon in that MNOM pool that we don't see. Okay. And so this made us think about the possibility of something called carbon metabolite repression. Okay, so what is carbon metabolite repression? Okay, so this is something that's relatively well known in the environmental engineering world, especially with regard to degradation of organic pollutants. Okay, and what's been shown is that if cells are in sort of very oligotrophic, low carbon environments, they tend to actually be better at degradation of like xenobiotic organic pollutants. And the theory is that if the cells are experiencing these sort of like starvation conditions, they'll start to actually overexpress a lot of their transport channels, overexpress a lot of their metabolite enzymes because they're just sort of desperately looking for and I'm describing a lot of sort of like personal motive to the cells, but obviously it's such a sort of like physiological response, okay? And what's also been shown is that if you then increase the labile carbon concentrations, the efficiency of micropollutant degradation tends to decrease. Okay, and the idea is that if they're in an environment replete with carbon substrates, replete with food, they don't need to devote as many of their resources to actually acquiring that food. Okay, so we were basically interested in testing whether this theory could explain some of these variations in arsenic. Okay, because arsenic is taken up through these GLPF transporters that also take up low molecular weight carbon substrates. Okay, so could there be some like interaction here between kind of like the DOM pool, the labile fraction of the DOM pool, and indirect effects on arsenic uptake? Okay, so here's basically the question here. Does carbon metabolite repression play a role in regulating microbial arsenic transformations through control of arsenic uptake through transporters that principally take up carbon chains? So we prepared DOM solutions with a range of concentrations from this Mississippi NOM and also from a rice straw leachate to try to look at DOM relevant for the types of DOM inputs into a rice paddy environment. And we used the E. coli biosensor method as well as looking at the expression of the GLPF transporter using RT-PCR. And so we saw here with the Mississippi NOM, sort of consistent with what we expected from before, as we increase the DOM or DOC concentration, if this is showing the arsenic uptake rate, we see this decrease in the arsenic uptake, where at very high DOC concentrations, actually uptake is completely inhibited. And for the rice straw leachate, we see a similar trend, but the effect is not as significant as what we saw for this Mississippi NOM. And in both cases, it's important to note we see DOM is stimulating sulfur. Okay, so as an indication that there's a lot of biocarbon. And then when we looked at the expression of the the GLPF transporter, is basically how actively are cells making more of these cell membrane or membrane transport channels. You can see here, there is a repression of GLPF expression in the presence of MNOM. Rice straw leachate, we see a somewhat different pattern where initially you see an increase in GLPF expression, but then at these highest DOC concentrations, you do see this strong repression in that expression. And it's notable that we have this sort of significant jump in cell growth at these two highest DOC concentrations is the same DOC concentrations where we see the most significant inhibition or repression of the transporter. Okay, so just from this one example, there does seem to be some connection between sort of like to what extent is growth stimulated and to what extent is the GLPF expression sort of like that. Okay, so yeah, so higher DOM represses GLPF expression, and repressed GLPF expression is also associated with less biosensor determinant. Okay, so we wanted to take it one step further and also look at methylation. Okay, everything I've talked about so far has really been about just uptake. How does arsenic get out into cells? How quickly does it get into cells? Does this translate into effects on actually production of methylated arsenic? So we use this cell, an Arsenicobacter roseanei, which was isolated from rice paddy soil. And this cell lacks the efflux pump. So most cells will take arsenic up and then they'll extrude it actually very rapidly to detoxify it. This microbe lacks this efflux pump. So what that means is the arsenite concentrations accumulate in the cell. And so it's a very efficient method. Okay. So we kind of use this as a tool to study sort of what might be affecting production of methylated arsenic because it methylates a lot. So it's kind of easier to see. And then we use that with HPLC ICP-MS to look at the changes in arsenic association. And so basically, we then using this Arsenicobacter roseanei, performed this biomethylation assay as well as looked at expression of the RSAM functional gene for arsenic methylation. And so for natural organic matter, so really nice consistency with our earlier results. Okay, as DOC increases, this is the methylation efficiency. So this is like the fraction of arsenic that's methylated, normalized by OD600. Okay, so we're trying to account and normalize for differences in cell growth that might result from different levels of dissolved organic carbon. So we see, a decrease in production of methylated arsenic species as DOC increases, which is sort of consistent with what we've seen before. And we also see a decrease in the RSAM expression. Okay. RSAM expression is induced by the intracellular arsenic, so it makes sense that if arsenic is not getting into cells, then RSAM expression will also be done. So all of this is telling a kind of consistent story. And thinking about the biosensor results, right, we're similarly seeing where uptake is shut off as we're measuring the biosensor, we're also seeing basically no methylation. Okay, so the results with the rice straw leachate are not as sort of consistent with what we saw before. Okay, what we see with methylation efficiency is an initial increase followed by a sort of possibly decreasing trend as we get to higher DOC, but not meaningfully returning to the level we saw in the control. Similarly, when we look at the expression of RSAM, we see this kind of increasing and then decreasing trend. Okay, so what we're seeing here is the form of the DOM has an okay. This Mississippi NOM is having a more significant, more clear inhibiting effect on leachate. And so some main points here. We saw that higher DOM led to repressed GLPF expression with arsenic uptake. And then at least for the MNOM, we saw a decreased expression for the rice straw leachate as part of a mixed bag. And we're actively now working with other sources of dissolved organic matter to try to understand how does this process occur with DOM with different diversity. And just kind of like to wrap things up here, here's kind of like a conceptual model. The idea or the theory is that with low biolabile carbon, the cells will express more GLPF transporters in order to acquire more, low molecular weight carbon substrates that will inadvertently lead to more arsenic uptake. In contrast, there's a lot of carbon, less dedication of resources to taking up carbon, right? Leading to fewer pathways for arsenic to enter the interstitials. That's the theory that we have some support for and we're continuing to test. So I presented this for the first time over the summer at the Goldschmidt conference, where there's a lot of interest in arsenic methylation, and you can see a lot of people in the audience just like, but we know that organic matter tends to stimulate arsenic methylation, not repress arsenic methylation. So what you're saying is really contrary to a lot of established knowledge. And so I think the point I just want to make to kind of put this into context is that I think our message is that arsenic uptake per cell, we would expect to decrease as biolabile organic carbon increases if our theory is correct, okay, this hypothesis is correct. But we also know that if we have more biolabile organic carbon, we'll also have more microbial biomass, okay? So we have these two different sort of like factors that are sort of acting in contrary directions. So like, what is the net result? It's quite likely that at low carbon concentrations, we might see this stimulus of arsenic methylation. But then what happens? Do we see a plateau? Do we see an increase and then a decrease? I think this is sort of like the question that we're now trying to understand. Interestingly, when I was at the Goldschmidt conference, I saw a presentation where someone was presenting data exactly like this, where they saw, they were looking at a chronosequence of paddy soils from China with radiant and DOC concentrations. And basically they saw as DOM increased in the paddy soil pore water, they saw an initial increase in the arsenic methylation followed by a plateau. Whereas a lot of the traditional thinking would be that you could sort of continue to see this increase due to stimulus of microbial growth and microbial. And they had just gotten their paper rejected because this pattern was sort of inconsistent with any prior knowledge, and they had no explanation. So we had a really nice conversation about that afterwards. Okay. So I'm running out of time here, so I'll just briefly run through the last few slides, which is to say that, so a lot of what I've talked about so far is about arsenic methylation, a lot of the research is about arsenic methylation, but that's not really the whole story, right? We also have to think about arsenic methylation. So when we talk about, what's leading to high concentrations of DMA, traditionally people have thought we have to understand methylation, we have to understand expression of arsenic, we have to understand this. Maybe that's totally misguided, right? Maybe it's really about demethylation, right? Maybe demethylation is actually controlling DNA concentrations. Okay, but it's not really about methylation, it's probably a combination. Both. There was a finding here of a new arsenic gene, ARSI, which is a carbon arsenic lyase. This it requires molecular oxygen. It's a dioxygenase. Okay, so maybe this explains what's happening in the alternate wetting and drying. Why, when we dry the soil down, you see a decrease in the methylated arsenic, but not the inorganic arsenic. Okay, but this is less relevant for flooded soil conditions. So we sort of accidentally came across this interesting finding where we were using metabolic inhibition, inhibiting methanogenesis to try to understand do methanogens play a role in arsenic methylation. Okay, so we use this methanogenic inhibitor called ES. Okay, and what we saw here was that when we used BES, okay, so when BES was used and methanogenesis was inhibited, we saw the DMA concentration sort of continuously increase. Okay, when methanogenesis was not inhibited, DMA increased and then decreased. Okay, so this we were thinking, okay, could methanogens be involved in arsenic demethylation? Actually, right? Does that make sense? Okay, so about the time my student first showed me this graph. It was a really beautiful study in ISME that I'm super jealous of, where they use a 13C labeled methyl group on DMA and track that 13C methyl into methane, okay? Which suggested indeed methanogens are somehow utilizing the carbon in this DMA methyl group as a substrate for methanogenesis and converting it to methane. Really nice, great study. And it is sort of like consistent with what we found sort of separately. So basically, right, the question now is DMA a carbon substrate for methylotropic methanogenesis? Methylotropic methanogens utilize methyl groups in small compounds like methanol. This is dimethylamine, also called DMA, to make things more confusing. Trimethylamine, okay. And so why not arsenic? Why not DMA? You know, could the methyl group of this DMA also do this? Methylotropic methanogenesis. So Hyun developed and executed this really nice study where we added TMA with DMA to sort of test whether stimulation of methylotropic methanogenesis could lead to enhanced demethylation of DMA. So we sort of grew up this methylotropic methanogenic community by adding TMA periodically. Once we saw an increase in methane production, we call this day 0. Dimethyl arsenic was spiked, and then we monitored DMA demethylation, and we also took mRNA samples periodically to look at expression. And so I'm really running out of time. So cutting to the chase, what we see again is that when methanogenesis is inhibited, DMA concentrations are fairly stable. Okay, when methanogenesis occurs, we see a decrease in DMA, and in the community that was sort of stimulated with trimethylamine, we see an earlier onset and a sort of faster demethylation, although at the end they sort of converge to genetically similar levels. We see an increase in inorganic arsenic, okay, so sort of showing that this is demethylation reaction. And then finally, a new postdoc in my group, Michael, who started about a month ago, he's been able to work with some of the sequencing data that we got and shown that, in fact, we are seeing a stimulus of methylamine utilizing, or sort of the methyltransferase is used by methylamine, methylamine utilizing methylotropic methanogens, okay, suggesting that we did effectively stimulate that community, where we also saw enhanced demethylation of DMA. So there's a lot more to come here. I got this figure. Michael sent this to me yesterday. So we're just getting started in analyzing omics data and comparing it to data. So the last point here is sort of thinking about new perspectives on how DOM may impact microbial transformations of arsenic. Sort of one new role here is the role that DOM can play in stabilizing iron-free colloids. Okay, and arsenic can partition to that colloidal phase, okay, to limit its uptake into cells. DOM, that is bio-labile, can inhibit cellular arsenic uptake through a catabolite repression-like mechanism. That's our theory that we're testing. We have some support for this, but we're not seeing all of our DOM showing that so convincingly. And then finally, you know, thinking about the role of sort of methyl-containing carbon compounds, methanol or methylamines as substrates for methylotropic methanogens that may also then contribute to or stimulate that. Okay, so that's it. Thanks so much for coming. Thanks for your attention. I'm happy to take questions. I just want to quickly, acknowledge funders and also the 4 students and postdoc, two postdocs and 2 students who are the main people who actually did most of this work, including Hyun, who's back there. It's done a really awesome job. So thanks again. Happy to take questions.
This has been a production of Cornell University, on the web at cornell.edu.